Hello everyone. In this video, we are going to learn about most probable number technique, which is also known as MPN test. It is used to check the portability of water. Water is the most essential compound found on the Earth's surface. In water, there are thousands of microbes present. So, if contaminated water was drunk by people, they are affected by bacterial infection like cholera, thyroid fever, bacillary dysentery, diarrhea, etc. Essentia coli is a fecal coliform bacteria mostly available in water. Most probable number technique is a test to use to estimate the concentration of viable microbes present in the water sample. MPN test is the best method and most commonly used to apply for the water test to check the potability of water quality. So, to ensure whether the water is safe or not. The presence of few fecal coliform bacteria in the drinking water could indicate that the water does not contain any disease causing agent. If many number of fecal coliform bacteria present in drinking water, it may contain disease-causing organism, so it is unsafe to drink. Principle of MPN. MPN is a statistical method used to estimate the viable number of bacteria in a sample by inoculating in broth. It is based on the principle of extinction dilution. Here, in the group of coliform, both aerobic and anaerobic bacteria were present. Bacteria that ferment lactose with gas formulating in 48 hours at 37 degrees Celsius. In NPN test, multiple tube fermentation approach to estimate coliform bacterial population. There are three steps involved in MPN test. Three steps are presumptive test, confirmatory test, and complete test. These are the materials used to check the water sample in the MPN test. For presumptive test tubes, Durham's tubes, pipettes, BGLB broth, that is brilliant green lactose bile broth or McConkey broth. For confirmatory test, ecosin methylene blue agar, petri dishes. For complete test, gram staining kit and other lab wares. Procedure for presumptive test. Take required glasswares. Prepare the media for single strength lactose broth and for double strength lactose broth. Pour 10 ml of single strength lactose broth in the 10 test tubes and 10 ml of double strength lactose broth in 5 test tubes. Add the diagram's tube in inverted position to check the gas formation. Sterile the media properly. After autoclave, inoculate the water sample with the sterile pipettes. By adding 0.1 ml of water sample in 5 test tubes of single strength lactose broth, 1 ml of water sample in another 5 ml of single strength lactose broth, and 10 ml of water sample in double strength lactose broth. Incubate the test tubes in the incubator. After 24 hours, you can check for results. For better results, you can incubate the tubes for 48 hours also. How to check the result? See this image. After incubation, the color change will appear. In 10 ml of test tube, 4 positive reaction. In 1 ml of test tube, 2 positive reaction. And in 0.1 ml test tube, there is no reaction. So the result is 420. Now take the standard chart. This is a standard chart. Check the MPN index for 100 ml. For 420, the MPN value is 22. So, per 100 ml of water sample, 22 coliform was estimated. This image shows the example of positive tubes of presumptive test. After the presumptive test, take the positive tubes for the confirmatory test. Take loop full of culture and stick it on ecosin methylene blue agar plate. After sticking, incubate the plates in the incubator at 37 degrees Celsius for 24 hours. If greenish metallic sheen is observed, presence of Escherichia coli. This image shows that growth of bacteria in EMB agar plate, but absence of metallic sheen. So, for complete test, take a colony from EMB agar and make a smear on slide for gram staining. This is the procedure and method of MPN test. Thank you for watching.